As you learned in the last lesson, reproduction is necessary for the survival of a species. There are two major reproductive strategies used by living organisms: asexual reproduction and sexual reproduction. You can probably guess that sexual reproduction involves two parents. Each parent is responsible for contributing half of the genetic material to the offspring. Those offspring are genetically different from each parent and from each other. Asexual reproduction begins with the prefix a, meaning without. Asexual reproduction, therefore, involves no sexual contact between individuals of a species and occurs by cell division. Asexual reproduction involves only one parent, and through the process of cell division, results in two daughter cells that are genetically identical to each other and to the parent cell. Organisms like bacteria reproduce asexually through a process called binary fission. This simple form of cell division involves copying the genetic material and dividing the cytoplasm. More complex organisms conduct asexual reproduction by mitosis. This unicellular protist reproduces by mitosis. Some plants, and even some animals like sea stars, for instance, can reproduce asexually by mitosis. Let's take a moment to review how the process of mitosis occurs. The cell begins its life cycle in a state called interphase. At this time, the cell is going about its everyday business, performing metabolic reactions and growing. When it is ready to divide, the cell copies its DNA. This step is important so that when the cell divides in two, each daughter cell will have one full copy of the DNA to store in its nucleus. After the DNA has been copied, the cell is ready to divide. Mitosis is the orderly sequence of events that take place during cell division that ensures that the two daughter cells are exact copies of the parent cell. Mitosis is divided up into 4 stages: prophase, metaphase, anaphase, and telophase. Let's look and see exactly what happens during the cell cycle. The cell spends most of its life cycle in interphase. At this point, the DNA is loose and unraveled in a form called chromatin. The cell grows and copies the DNA. Mitosis begins with prophase. During this stage, the loose chromatin condenses to form chromosomes. 2 identical chromosomes are attached by a centromere, and the pair are called sister chromatids. While the DNA condenses, the nuclear membrane starts to break down and disappear. The centrioles are activated during prophase, and spindle fibers begin to extend outward toward the chromosomes. The spindle fibers attach to the centromeres of the chromatid pairs, and the centrioles coordinate their movements as if they were puppets on a string. Plant cells do not have centrioles, however, the movements of the spindles and the chromosomes are the same. Cell division progresses into metaphase as the centrioles guide the sister chromatids to line up along the center of the cell. Anaphase begins as the spindle fibers pull the sister chromatids apart, and one copy of each chromosome heads towards opposite ends of the cell. The cell itself begins to elongate. The final stage of mitosis is telophase. The daughter chromosomes reach the ends of the cell and begin to unravel back into chromatin. A nuclear membrane will form around each group of chromosomes to make 2 nuclei. The center of the cell pinches inward, forming a furrow. In plants, an equatorial plate forms between the 2 new cells. This plate will become a new cell wall, separating the cells. Mitosis is now complete, but cell division will not be complete until the cytoplasm has completely divided in 2. The process of dividing the cytoplasm is called cytokinesis. At the end of cytokinesis, we are left with 2 daughter cells that are identical to each other and to the original cell. Both of these new cells will immediately enter interphase and remain in this state until they too are ready to divide. Here is an overview of the cell cycle, beginning with interphase, cycling through prophase, metaphase, anaphase, telophase, and cytokinesis, and ending with two daughter cells. The fact that this is a continuous cycle for cells is why we refer to this process as the cell cycle. As you just saw, asexual reproduction occurs by cell division, either simple binary fission or the more complex process of mitosis. Asexual reproduction produces genetically identical offspring. Sexual reproduction, on the other hand, requires two parents to contribute genetic material, so the offspring are genetically unique. In order to pass on their genetic material, the parents must package their chromosomes into sex cells or gametes, egg and sperm. When egg and sperm fuse, a new individual is created. Egg and sperm are generated by a different kind of cell division known as meiosis. Meiosis is somewhat similar to mitosis, but with some very important differences. Mitosis has one division that results in two identical cells. Meiosis, on the other hand, includes two divisions. The first division results in two cells. Then those two cells divide again in the second division to make four cells. In order to understand the necessity of the two divisions in meiosis, you have to understand a little about genetics. All our genetic traits are coded in DNA. That DNA gets coiled up, and coiled up, and coiled up again to form a dense body of genetic material we call a chromosome. There are a number of different chromosomes housed within the nucleus of a cell. Each chromosome carries the genetic code for numerous traits. The number of chromosomes that an organism has in its nucleus is very specific. For example, a fruit fly has 8 chromosomes in its nucleus, while an elephant has 56. This fern has 184 chromosomes, and this chicken has 78. The number of chromosomes that an organism has in the nucleus of its cells is called the diploid number. 2n represents the diploid number. We as humans have 46 chromosomes in the nuclei of our cells, so our diploid number is 46. It is important during sexual reproduction that the organisms maintain the correct number of chromosomes. If a man were to package all 46 of his chromosomes into each of his sperm, and a woman packaged all 46 of her chromosomes into her egg, then when the egg and sperm fused during fertilization, the resulting cell, called a zygote, would have 92 chromosomes, twice the amount of chromosomes it should have. The generation after that would have 184 chromosomes, and so on. With each generation, the diploid number in humans would double. But this does not happen because of those two divisions that happen during meiosis. The two divisions of meiosis cut the diploid number in half. This halved number of chromosomes is called the haploid number and is represented by the letter N. In humans, the haploid number is 23. Because of the two divisions of meiosis, a human sperm has 23 chromosomes, and a human egg has 23 chromosomes. When they fuse during fertilization, the resulting zygote will have 46 chromosomes. This zygote can then develop into a healthy human baby who received half of its genetic material from its mother and half from its father. Now you understand why meiosis includes two divisions, and you can see that these two divisions result in 4 cells. Mitosis, on the other hand, produces only 2 cells after a single division. The 2 cells produced by mitosis are genetically identical to each other and to the parent cell. They are called diploid because they each have a full set of chromosomes in their nucleus, just like the parent cell. In contrast, the 4 cells produced by meiosis are genetically different from each other and from the parent cell. These 4 gametes are haploid, meaning they contain half the genetic material of the parent cell. This is important so that when fertilization occurs, the resulting zygote has the appropriate number of chromosomes in its nucleus. Mitosis is performed by all cells of the body so that an organism can grow or repair damaged cells. Meiosis is performed only by cells in the ovaries of women and the testes of men. Meiosis occurs specifically to produce egg and sperm for reproduction. You can see from this list we have created the differences between mitosis and meiosis. These differences are important for the different functions that these 2 types of cell division perform.